Hello friends, good morning and welcome to the lecture 1 of week 8. So we have looked at various assembly approaches and methods of sequencing. So repeats pose one of the important challenges when we are sequencing a genome. So in this lecture we will look at what is the significance of repeats, what are the different types of repeats, what is their respective composition of the genomes that contain these repeats, and how we can use a sequencing approach to deal with it. Okay, so basically repeats is an integral part of genomes. Even like prokaryotes contain some repeats, even if they have a requirement to keep as much genic region as possible and keep it compact genome, but still they contain some repeats. And plants contain another extreme of it, which contains almost 90% of genome could be repeats in some cases, right? So repeats are also of multiple types, simple and complex, and they pose challenges when we are sequencing as well as assembly, and also to plan how much amount of sequencing we want to do. Okay, so like in the case of human genome, for example, you can see that if I say 100% only 25% is genic and 75% is intergenic, and this 75% also like you can say that 55% is repetitive, of which the simple repeats are 10% and interspersed repeats are 45%. Okay, like these are in majority, whereas this is like 6 to 8%. So that means repeats of different types constitute a large component of the genome and needs to be correctly characterized. Okay, so in this picture also you can see that basically the genic region is like less than 2%, whereas signs, lines, transposons, these constitute a lot of regions that which are repetitive. So one is tandem repeats. That means tandem means repetitive. For example, in this case, like if I say TG, 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 so this TG unit is repeating, and like every third base pair is TG here, T here, and G here. So tandem, okay, occurring again and again, right, repeating fashion. So this could be like as simple as dinucleotide like TG, or CAG trinucleotide, or even CNG, N can be anything, and CTG, CAG, CGG. These are the triplet repeats that are involved in the several trinucleotide repeat disorders, right? So their presence and copy number and the location of their presence is also important for the analysis in the genomic analysis. And so like this is one sequence, and you can see that there is a stretch of repeating TG or GT units. So you can read it as GT or TG, right? And these are short sequence repeats or simple sequence repeats, but they do not they never exceed more than like 30 to 40 units, right? And okay, so they may not cause that much problems in sequencing, but still for short reads they may cause some problems. Okay, then there are telomeric repeats which are at the ends of the chromosome, and when you are doing a chromosome like telomere to telomere sequencing, as the term says, right, it's a complex task, and that is why human genome also took a lot of time, right, in deciphering these kind of repeats. And the telomeric repeats have a important role in keeping the structure of the chromosome intact, and also they also play an important role in aging. Okay, then there are mini satellite DNA, which is like 6 to 100 nucleotide repeating units, generally like GCRH. Okay, and they have a high mutation rate, and also show like a lot of diversity in populations, because in terms of their location as well as repeating units. Microsatellites, like as I mentioned, like simple sequence repeats or short repeats, tandem repeat, STRs. Okay, and they are also like mini satellite, but and also have high mutation rate. Like example is like TG, or CAG, like that. Okay, so generally you keep it right tick like this, CAGN. So that means there is a repeat which is CAG and repeating n number of times, n is in the subscript. Okay, and generally microsatellites have been used to prepare the microsatellite maps of the genome, and also to determine the paternity, and also in case of some forensic analysis, that because these repeats are conserved yet polymorphic. Okay, then there are dispersed repeats which jump here and there in the genome and cause disruptions in the coding regions or starting region of genes, and that is how they increase their numbers, and they could also get inserted by various mechanisms, right? And in eukaryotes, they are comprise a lot of region in the eukaryotic DNA, right? And so basically, like in case of maize, there was experiment by Barbara McClintock where you could see that that she first told the presence of transposons in the genome. Okay, so signs and lines are two kinds. I'm just doing a repetition of what initially also in the previous classes I've covered this, but I'm just giving an overview of repeats so that I can connect the next part to it. So you can see that signs and lines are two interspersed repeats, short and long interspersed repeats. So LU repeats are very common in the human genome, and their insertion and location with respect to genes is very important for the gene expression, right? And similarly, there are LTRs, which is like almost 21% of human genome is composed of it. So these are two of the major components of primates or human genome. Okay, and basically these LTRs are distributed throughout the genome and have a lot of functions, such as because they are random insertion here and there in the genome, they can cause disruptions, and otherwise if they are not lying in a region of any function or known function, they be called as inactive elements, and they cause disruptions when they get insert in the various genes like colorectal cancer, or also because of their random insertion they can also lead to evolution of new genes, right, or change in the genes, right? And function of lines and signs is not fully known, but they are like integral part of the genome, and must be playing an important role in the evolution of humans and other eukaryotic genomes, right? So one of the commonly used tool to determine this repeat content, which you should accurately determine, which is a part of your genomic analysis, because you have to report these numbers to tell that how much of your genome is repeated, what kind of repeats are more frequent, okay, which also tells a lot about its function and possible implications on various aspects of the genome phenome or genotype phenotype analysis, right? So accurate and correct determination of repeats is very important, and RepeatMasker is one of the software which also has an integral database of repeats from various genomes, and you can also improvise that based on when you are doing the analysis, right? It uses repeat library files for the annotation of repeats, okay, and that is important that you select a closely related, or if the genome is already known and you are doing resequencing of that genome, so using a correct or close reference file, repeat file, right? So and basically to map the repeats and to because it is using a library of repeats and mapping on your genome and then masking your genome, it is using various programs such as RMBLAST, crossmatch, NHMR, which is hidden Markov based, or ABB BLAST, so which are different variants of BLAST basically to do quick and faster mapping of repeats and annotation of repeats on the genome, right? And it can be used both command line mode or the GUI mode, right? GUI mode has some limitation that you cannot upload more than like I think 100 KB, but in case of command line you can have the library and the software installed in the system, and you can run larger genomes also. Otherwise in the GUI or like web based, when you submit a query you have some limitations, okay. So this is the web page for the GUI, so you can like choose a file or paste your sequence here. Generally most of the programs are like that, okay. Choose the program that you want to use, okay. By default it is RMBLAST, which is maybe the default and appropriate method. You want to do very quick fast or basically default slow annotation, which is you can decide, but default is okay generally. You have to find the library, so you have to should choose what organism you are doing so that it can pick the appropriate closed library. Okay, how do you want to return the files in HTML or a compressed format, tar. Okay, and you should tell your email address because it may take some time to do the repeat masking, and your job will be returned by email, a link, and you can download the information from there and you should submit. And basically you can also include other options like do you want alignments of repeats and you want the resultant DNA to be masked. That means from a chunk of your sequence, the regions that are repetitive will be written as n. So repeats will be converted to n so that they don't confuse your gene prediction or other analysis later. Okay, and then contamination check and what type of repeats you want to find, both interspersed and simple repeats, only simple repeats or only complex repeats. Okay, for simple repeats there are other programs also, or if you want to do only simple, but I think for the complex repeats, RepeatMasker is one of the gold standard of finding the repeat. Okay, and then if you want to do this locally, then you have to download the RepeatMasker. For example, the latest version is 4.1.5. you should download a tar.gz file, which is a compressed and zipped format. Okay, tar and zipped format, and you should take it to the folder that where you want to install it, like user local. Okay, move to that folder, go to that folder, unzip the files. Okay, and this will unzip this file, and then you should untar. That means because it's a bundle of several folders and files, you should untar that tar xff. This we have done earlier when we are doing the Linux part. Okay, and then you have to basically also download the libraries from the site. Okay, and also put that in a defined folder so that the program do not face any issues while it is looking for the library file. And also you should always read the readme file or install file that comes with the program, any program that you download, because that tells you information about what to do, how to do the installation, the steps for installation of a program, and also to place the appropriate files at various paths, and also to set the paths, right. And then RepBase, which you can also download and keep it in the same path, right. Same steps would to unzip and untar it. Okay, basically I'm not going details of this installation because we have already covered that in the previous classes, right. Then for installing, you should go to the RepeatMasker, the paths, change directory, the path, and then perl./configure. So it will configure the program for your system and basically install it. After that you can use. So now RepeatMasker can take multiple formats, and this multi-faster format like this, where there is first line and is annotation and then followed by sequence, and this is the default program, default files type. Okay, faster format. And then you should tell the other options, like we have seen in the GUI option, that how you want the data, what genome it is, and what library you should use, right. And it will give you the output, like for example, this your input was a file that contain all nucleotides and this ends were absent, right. Now whichever place the repeat was present, it has placed ends. Okay, so now if you want to gene prediction or do some calculation of, say finding some mutations, or right, so basically, or the region which is not repetitive, then basically ends will not interfere in your those calculations, right. It will give you a summary of information, like this is annotation of the mask file, that what is the score, person identity divergence from the reference repeat, right, where it is present, start, end, right, which repeat family it matches to, right, and what is the status of annotation of that repeat family. Sometimes it is known, sometimes it is unknown, right. Gypsy elements, LTRs are known, for example, right. And where in the repeat they are matching, like start and end similarly, right. and Smith and Waterman SW score for the match, which tells that which is basically the BLAST score. You can tell, say that similarly you got in BLAST also, that how much is the score of match, okay, and the percent substitutions in the matching region, because it is not that exact region will be matched. There will always be some substitutions or changes in with reference to the query and subject. Okay, percent deletion, percent insertion, position start, and I think I explained all these things in the previous one also. So it gives you comprehensive information of where the repeat is exactly lying, how much is different from the reference repeat, the changes that you have found in your repeat in terms of percent deletion, insertions, and identity, right, and its score also. And basically the default output is kind of acceptable output, and it also gives you a summary of like this summary table, where it tells that okay, for the different elements we have just seen in the quick review of repeats we did in this class. So like signs, lines, LTRs, gypsy, how much is the relative, how much bases they are occupying in the genome, and their relative percentage, like 21% or 10%, right. And similarly for simple repeats, okay, it gives you okay small RNA and rolling cycles, and basically DNA transposons. So it tells you that how much bases, how much region they are occupying in the your piece of DNA or genome. Okay, it also gives you an alignment file of the repeat and the genome, so that you can use that to design primers to see the regions which are non-matching with the repeat, so that you can amplify those regions for further experimental or computational analysis, right. So RepeatMasker is a default or important program to first identify the repeat, annotate it, okay, identify that it is present. Annotate means that what kind of repeat is present, their respective numbers, composition in the genome, and also to mask that repeat in the sequence so that they don't interfere with the further analysis or gene prediction and other annotation objectives that you have. Okay, for example, in case of human, almost 56% of the genome was found to be repetitive, right, and it was done by RepeatMasker, and this program is available at repeatmasker.org. It's a very old program, I think more than 20 years old, and it's one of the gold standard and important program that you should use. Similarly, for simple repeats, there is another program called Tandem Repeat Finder. It is also a standard old program and has been used in several cases to find tandem repeats in eukaryotes or prokaryotes. So tandem repeats are like simple short sequence repeats, right, and it can identify those repeats, display their start end in the genome or in the region that you have given as input, and also basically it can tell that okay what kind of repeat it is, and okay in the region of your the file that you have given. Okay, so for example, okay, how to install it? So basically you will download the program, then again it is a TRF 4.1 10.0 could be the latest version. tar-gz is the standard generally, because every program will be zipped as well as also contain a bundle of files, so it is tar also. Okay, so you have to uncompress it, also unbundle it. Okay, then go to the directory, make directory build. Okay, so basically all these instructions will be there in the readme file. If you look at after this doing the entire thing, you will get one file which contains the readme file, and that file will have the instruction of installation, or there could be install file. Okay, these will be text files, right. So go to the folder of the program. Okay, make directory build is the folder that contains all the executables. So go to that cd build /configure make install. Okay, and for make install you need sudo, that means you need root access or administrator access. So we have already covered these things when we were doing the Linux and installation of software, right. So generally most programs are like that, have a standard way of installing, and okay, sometimes there could be some dependencies and it will prompt you that okay, this file is missing or this error. So one way is to get that file and install. Other is like you can also look at the Google, you can search for some previous errors which people have reported like that earlier, and then what is the solution for that, and this way like you will learn how to resolve problems of installations and using these. Okay, then you can use the program. It's simple like command line program. So you can give an input file and other parameters where these are the different parameters that you can give, like okay, what is the mismatch penalty, indels, insertion deletions you want to allow, minimum score or minimum threshold for which you can accept a repeat or to report that, okay, whether you want a masked file as the output, okay, and the flanking sequence of the repeat, right? So like again, like this can take FASTA format. As I mentioned, FASTA format is one of the standard format that is used by almost all the programs, because generally the programs need the sequence and some annotation for that, right? And then it will give you the output, okay, where you can see that the repeat which was present is now masked as n's, right? So like the repeat was a TA, like T A, you can say A, GC, TA, G, GC, TA, G, GC, TA, GC, TA, GC, TA, okay. So basically GC, ATA, GC, ATA, like that. You can see it's a repetitive region and it has masked it, right? And also it will give you another text file that contains the further information about the repeat, that what is the consensus repeat and what is the your repeat that you have found, okay, in the repeat sequence, okay, like this here mentioned here, right? So basically these options will help you to do further analysis with the repetitive region of of your interest, if you are interested in some kind of repeat. But overall summary is something that generally you may like to report in the genome paper, okay. There is another EMBOSS is a suite of several programs, bioinformatics programs, and finding repeats is one of the program. We have also done one class in EMBOSS, okay. So eQuick Tandem is one of the program available within EMBOSS, which can be run in command line as well as GUI mode at the EBI site. And when you download the package and install, you can find eQuick Tandem there, okay. So it can identify the regions that contains repeats, right? So right, and not the repeat itself, but also the region that contains those repeats are likely to contain those repeats, okay. And this is one of the program that you may like to use to reconfirm or basically check a region for the presence of repeats, okay. So this is the web-based version of this, which is say available here, okay. Upload your sequence or paste it here, and specify the parameters like maximum repeat size you want to see, threshold. Generally defaults are okay, and your email address, and just run it, okay. And it will give you the output something like this, that where the repeat is present, start, end, and the count of the repeats inside the sequence. And similarly, in addition to eQuick Tandem, we also have eTandem, which tells you exact tandem repeats in that sequence, right? So it tells the type of repeating unit and its number of repetitions in the genome, right? Again, you can use it web-based or install locally, okay. And it will give you the like what repeat it has found, what is the identity of the repeat with respect to the reference repeat, right? Their start and that means location in the genome that you have, or the sequence that you have given, which strand of the sequence that is present. It checks both the strands, forward and reverse, plus and minus for that, okay, and the score for the repeat, okay. So just take a look at like in different genomes what is the repetitive content. For example, in case of peacock genome, if you see right, which is a bird genome, you can see it has signs, Penelope, and sign lines, okay, sign line, okay. So basically all these repeats are there, okay. They occupy considerable number in the genome, right, but lines are the most frequent, okay. Now in case of plant, if you see, so basically almost 64% was repeat turmeric, right, and signs and Penelope was absent, and lines was one of the common repeats you can see in the case of plants, right. In case of animal, it has all the 3 element repeats like tiger, line, LTR and signs, which are commonly found in these kind of animal genomes, right. So different type of organisms will have different portion of repeats, and sometimes you can, when you look at this file, you can find some repeat which is like over represented or too much, so which could be one of the finding of the analysis and is reportable finding, right. So you should look at those observations very carefully. Okay, so for example, in case of aloe vera, which is 16 GB in size compared to like 0.5 GB of say banyan tree, 32 times more than that, and it is more because it also contains like almost 82% of that genome is just repeat, right. Similarly, in case of turmeric, 64% of the genome was repeat, right, of which like 62% was interspersed repeats, and like just 1.5% was tandem or SSRs, right, simple repeats, right. And in case of bird, blue peafowl or peacock, blue peacock, almost 10% region was repeat, and genome was also compact, only like 1 GB in size, and it contains like so many genes and other regulatory regions, right. So the amount of repeats vary in different genomes, and it is important to estimate repeat and from the genome that you have got. And also, if you have a if you know that okay, you are going to deal with a plant genome, then you should be prepared for long reads more, so that you can cover these repetitive regions. For example, like if this is your genome and basically this is a repetitive region, okay, if I do short reads here, okay, so all these reads which are coming from this region, right, are exactly identical or almost identical, and I will not be able to see that connect these based on any overlap, because it will be like ATGC, ATGC or GACT, GACT or CAG, 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 right. So I will not know that how to connect the pieces, but in this case a long read will be helpful, because it may cover a region like this or the entire region like this in a single read. Okay, so that will have the repetitive region as well as the flanking region, so that I can accurately put that piece back into the assembly, so that I can cover that region of interest, and also correctly can tell how much is the repetitive region and what kind of repeat was present in that region. So that is how the long reads play an important role in the hybrid assembly. Okay, these are some of the papers that you may like to see, or references that I have used in this work, in this lecture. Okay, so basically in this lecture we looked at what are different types of repetitive sequences, including the simple and complex repeats, and how to identify, annotate those repeats in the genome, and also their role in the genome, and their variation when we deal with different type of genomes. And this is very important information that you like to report in a genome paper, and also important for your genome sequencing and assembly, and also the analysis part. Okay, thank you for today.